Cyanobacteria are an important phylum of photosynthetic microbes that are increasingly recognized as useful platforms for synthetic biology and renewable biotechnology applications. So developing robust techniques to simplify the design, cloning, and introduction of heterologous DNA into different cyanobacterial species is important. So this technique demonstrates a well-established method called conjugation or triparental mating for introducing DNA into cyanobacterial species that are not naturally transformable.
Conjugation has been successfully performed in a wide variety of cyanobacterial strains, from single-cell to multicellular filamentous species. If you're attempting this technique for the first time, please take care to handle both your cyanobacterial and your E. coli strains with care. For example, do not centrifuge above the values given in the protocol, and do not vortex. Mixing bacteria and cyanobacteria may seem strange to first-time users. We hope that by demonstrating the conjugation process, we will help to clarify this.
After construction of level 1 assemblies, choose an appropriate level T acceptor vector. Choose an appropriate end link to ligate the 3' end of the final level 1 assembly to the level T backbone. To assemble one or more level 1 assemblies in level T, prepare a reaction mix with BPI1 and the required end link vector. Set up the thermal cycler program and proceed according to the manuscript. On day 1, grow cyanobacterial culture by setting up a fresh culture of Senecacystis PCC 6803 or Senecacoccus elongatus Utex 2973. With a heat